As you have seen, the UV light is damaging the DNA. It is the one of the major causes of DNA mutations. So how these UV lights damage the DNA? So mainly two major photo products account for nearly all of the UV induced damage to the DNA. So mainly two photo products are responsible for this damage. They involve the dimer formation between adjacent pyrimidine bases on the same DNA strand. So make it clear that the dimer formation takes place between two pyrimidines which are present adjacent to each other but on the same strand. So this is not a dimer formation between the compatible compatible bases which are present on the opposite DNA strand. So this is between the adjacent pyrimidine bases. The first pathway includes the formation of cyclobutane pyrimidine dimer or CPD. This is mainly responsible for the DNA damage, and it accounts for about 75% of all the UV induced damage. So the total damage caused by UV, the 75% of that damage is contributed by the formation of this CPD. The cyclobutane ring is generated by forming one bond between the carbon atoms of one thymine or pyrimidine, and the second bond is formed between the C6 atoms on the adjacent pyrimidine rings. So if two rings are present, pyrimidine rings are present adjacent to each other, so on these rings, first bond is formed between the C5 atom. So C5 atom of this base pyrimidine and C5 atom of this pyrimidine. They take part in the formation of one bond, and the second bond is formed between the C6 atoms of both these pyrimidine rings. So in this way, two bonds are formed. These are actually covalent bonds. So these two bonds are formed between two adjacent pyrimidine rings. So here you can see in this diagram, two rings are formed, one here and the other one is here. So these two rings are between C5 of both the rings, and the second bond is between the C6 of both the rings. So in this way a dimer is formed. This is called a dimer. So here you can see in another figure how these affect the conformation of the DNA. So here you can see between these two thymines this dimer is formed. This is normal DNA strand, and in this normal strand, if two thymine are present adjacent to each other, and if UV light falls on these thymine, this UV light falls on these thymine, they form dimer. So the formation of this dimer produces a small kink in the DNA strand. So here you can see this is a small band here. This band is mainly produced due to the formation of a thymine dimer. The most common cyclobutane pyrimidine dimer is the thymine-thymine dimer. So this is formed between two thymine bases. Cytosine-thymine and cytosine-cytosine dimers are also formed, but they are formed at comparatively slower rates. So the most prominent dimers are formed between thymine and thymine. Structural studies show that BDNA is a form of DNA that can accommodate a single thymine-thymine dimer, forcing the helical axis to bend by about an angle of 30 towards the major groove. So you might be remembering the major groove which is present in the DNA helical structure. So this dimer, it produces a bend of about 30 degrees and this band is towards that major groove of the DNA helix. The dimers 3' thymine can form a normal base pairing with its adenine partner on the complementary strand. So as you know, in this dimer 2 thymine are involved. One thymine is towards the 3' end and the other thymine is towards the 5' end. So the thymine present in towards the 3' end, it can make normal base pairing with its adenine partner, compatible base pairing on the other strand. While the interaction between the 5' thymine and its complementary adenine partner will be weaker than normal because a single hydrogen bond can be formed here. So normally between adenine and thymine, 2 hydrogen bonds are formed. Similarly, you know, between guanine and cytosine, 3 hydrogen bonds are formed. So here between adenine and thymine, 2 hydrogen bonds are formed. But here in case of dimer formation, the thymine present towards the 5' end, it makes only 1 hydrogen bond because the space for the formation of second hydrogen bond is not available. Thymine-thymine dimers are often used to study DNA repair systems because they are stable, because dimers are stable, they are easy to form and are easy to detect. So these 3 properties make them an excellent tool to study the DNA repair systems.